Bismillahirrahmanirrahim. Today we are going to discuss about how the polypeptide chain is formed from a transcript. A transcript, is formed by the process known as transcription. I mean, it is the part of central dogma. You know, uh, Central dogma is what the message which is there, which is inscribed in the DNA that is transferred for the function of any cell or subsequently an organism. You have already stated what the formation of DNA from DNA is known as what application, and formation of an RNA molecule from DNA is known as what transcription, and the formation of a polypeptide chain from RNA molecule is known as translation. Today, the topic of our discussion is translation. Now, you know, here is suppose this is what the DNA molecule or a gene. It undergoes the first process, that is what transcription, and as a result of transcription, what is formed? That is RNA. RNA molecule. You may know there are three types of the RNA molecule. Those are what? Messenger RNA, ribosomal RNA, and transfer RNA. In the last lecture, you have studied what? That a single polymerase enzyme is required for the formation of three RNA molecules in case of prokaryotic cells. In case of eukaryotic cells, there are three different polymerase enzymes required for the transcription process. So here is another difference between, you can quote it as the difference between the prokaryotic and eukaryotic cells. Well, this first process is known as the transcription, and this is the first part of central dogma. And this is the second part. As a result of this process, translation, what is formed? A polypeptide chain is formed. These you can say that these are what the amino acids, adenine, I'm sorry, glycine, and tryptophan, and methionine, and so and so. Okay. Well, as I was explaining here that transcription involves three processes, and these are three types of molecules are formed. Those are what the formation of rRNA, formation of tRNA, and formation of mRNA. So we should know what sort of these molecules are. The messenger RNA is a single stranded molecule, and this is a linear molecule. Okay. The transfer RNA is a clover shaped molecule. You can see here, this is what the transfer RNA. Okay. And it has this is what the 5' end, and this is 3' end. 3' end is open for forming the amino acyl linkage. How the amino acyl linkage is formed, I will discuss later. First of all, you must know the structure of this molecule. This tRNA molecule has three loops. This is known as DHU loop, or simply the D loop. Why we call it DHU loop? I have told you earlier, dihydrouridine loop. Dihydrouridine loop. And this is known as what? T-psi C loop, or T-psi loop, or psi loop simply. This is known as what? The stump. This one is known as stump. And this is ladies and gentlemen, this is the anti-codon loop. These three bases are actually the bases which play the crucial role during the protein synthesis. Well, the third one is what? Ribosomal RNA. The ribosomal RNA is found in smaller subunit of ribosome and larger subunit of ribosome. Okay. Well, now we should know how this machinery works. Whatever I was telling you people, this is what the machinery required for the synthesis of proteins are for the translation process. The machinery required for the translation is what? DNA, RNA, and there are three different RNA molecules, as I have told you people, and the enzymes. Okay. Well, suppose we take into consideration the first step. The first step is what? The formation of messenger RNA, and the messenger RNA comes out of the nucleus. Suppose this is what? Suppose this is the cell, and this is suppose the nucleus. Okay, the messenger RNA would come out of the nuclear pore. Okay, this is suppose the DNA molecule, and this is the transcript. It will come out, it will undergo certain processing, and after that it will come out. I will tell you people that this is known as what? The nuclear mRNA. Nuclear mRNA. And nuclear RNA molecule. And as it comes out, it is known as what? Messenger RNA. So here is the difference. I will tell you people, why do we call it the heterogeneous nuclear RNA, or simply the nuclear RNA, and why we call it this very molecule as the messenger RNA? I will tell you people in the next lecture. Well, in that lecture we'll study the regulation of transcription process. Today we are going to study only we are going to understand what is the translation process. Well, suppose we take into consideration the first step. That is what? This is the first step. I mean, the process involves the first step, and that is what? The arrival of the messenger RNA are the text sent by, or the code sent by what? The gene, or what? The governor of the cell. That is what? DNA. The second step is what? The formation of amino acyl transfer RNA. Amino acyl transfer RNA. This step involves what? The combination of the chemical combination of what? This is I have formed the diminutive of what? This molecule, this transfer RNA. You can form this small notion of this molecule. Well, this molecule, I have told you people, this is what the here is what? OH. Okay, at the 3' end, and this is what? The 5' end. At the 3' end would combine what? The amino acid. And here the enzyme is involved. As I have told you people, the enzymes are required. So here the enzyme is which is involved, that is what? Amino acyl amino acyl synthetase. Amino acyl synthetase. Well, by the combination of the RNA molecule and an amino acid, here the amino acyl bond is formed, and this whole structure, this one is known as what? The amino acyl tRNA. Now it is ready to find the codon. It has to reach the transcript. Okay, the first amino acid, ladies and gentlemen, which combines the first amino acid tRNA which is formed, that carries methionine. That carries methionine. I mean, the initiation codon of all the transcripts that carries what? The the codon for what? For the first amino acid to be uh, to form, to initiate the polypeptide chain, that is what? Methionine. So now we let us come to understand how, if you have the question, how the 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 transfer RNA molecule combines with an amino acid. You know, here the here is what I've tried to show you people. This is what the amino acid molecule, and here is what the 3 prime and 3 prime end. Oh, this is what one one two 1 2 3 3 prime end of what? The a molecule of transfer RNA. I told you people, this is 3 prime end, and this is again 3 prime end. Okay, so if you have understood, now let us come to this point here. This transfer RNA, it has this transfer RNA along with amino acid. We call this molecule as what? The amino acyl tRNA. This will reach here. This is the AUG. This triplet of nucleotide is what? AUG, adenine, uracil, and guanine. Ladies and gentlemen, always the initiation codon is AUG. Okay. Now you can see this. This is what the initiation factor, which brings the smaller subunit to combine with what? The amino acyl tRNA. Amino acyl tRNA and smaller subunit. This is the smaller subunit of ribosome. This one. You can see what this is, 40S. Okay, and 40S ribosome is brought along with the amino acyl tRNA. This is what? The amino acid. Okay, it is happy now, it is going somewhere. Okay, and this is what? The tRNA, and this is the initiation factor, this one, and this is what? The smaller subunit. Now this initiation factor reaches where? This initiation factor along with the smaller subunit combines this molecule, and both, I mean, the transfer amino acyl tRNA and smaller subunit of ribosome reach the initiation codon. Okay, clear? Now, now we suppose that then what is the next step? Here is the next step. This is the initiation factor. This is initiation factor. It brings what? The larger subunit. It combines here with the smaller subunit and what? The amino acid tRNA. And here three sites are formed. This is known as what? P site or peptide site. This is known as E site or exit site. And this is known as what? The A site. This one is known as what? The A site. This one. Okay? This site. Well, now you see the situation is that the methionine carrying amino acid tRNA is there at the destination or what? The initiation codon, or I mean AUG, and the largest subunit has reached the what you suppose now? The elongation. I mean the next amino acid should reach, and that should form the polypeptide chain. That would form what? The peptide bond or peptide linkage, and so on, so on, so on. Elongation would occur. Let me tell you, people, that this is I have supposed that this is what glycine carrying tRNA. We'll call it what? The next amino acid tRNA, and it has entered into this system through what? The A site. And what would be next? This methionine would form the bond with glycine. This is what? The peptide bond. This one. This is the peptide bond formed between methionine and glycine. This one is here. Okay, I mean the next amino acid has been coded. Okay, and this process will keep on until the stop signal reaches. Okay, there are the stop codons at the end of the transcript. Nature does not wish that there should be any sort of a mistake in the cell. Our cells, they speak. Our cells, they walk. Our cells, they form our body. Cell is a unit of structure and function. So if a cell commits a mistake, it means that that mistake would be expressed in the body of the organism. Okay, so the stop codon would reach here at the stop codon, and protein synthesis would end. This is what? The story of how the initiation, how the amino acid tRNA is formed, and how the initiation occurs, and how the elongation occurs, and how the termination occurs. So here I would like to summarize this process as what? The initiation, and what? Elongation, and termination. That is, termination is carried out by what? The termination codon or the stop codon. And there are three stop codons I have told you people earlier. Okay, and this is all about how the translation occurs. In the next lecture, we'll study how the regulation of formation of proteins takes place by transcription process. Okay, we'll discuss in the next lecture, inshallah. Thank you very much.